Receptor tyrosine kinases are a way for signals to conduct responses from within cells using proteins and phosphate. Tyrosine kinases are polypeptide chains made of amino acids and utilizes enzymes to phosphorylate. They work either in pairs or in groups. When a signal ligand attaches to the receptor in the external region, it causes nearby RTKs to be summoned. When another RTK is attached, dimerization takes place in which the tyrosine in the intercellular matrix is activated. The tyrosine will then cross-phosphorylate the adjacent tyrosine with a phosphate from ATP. Once all 6 tyrosines have a phosphate attached, they have become phosphotyrosine motifs. The phosphates have a high affinity to proteins with an SH2 domain, causing binding to each tyrosine. Different kinds of proteins that are performing signal transductions are able to attach, even at a single moment, so multiple cellular responses can take place. The proteins will then go through conformational change and eventually detach and continue with the cellular signaling cascade. Ligand-gated ion channels are cellular membrane receptors that regulate the flow of various molecular ions through plasma membranes. The ligand channel is made of integral membrane proteins and is located on the plasma membrane between the extracellular and intercellular matrix. When a signal, called a ligand, connects to the allosteric receptor site of the channel, there is a change in protein conformation that allows the gate to open. The gate of the channel only opens and closes as a response to a connection with a ligand in the allosteric site. This connection is also commonly known as a lock and key, meaning that the gate relies on a ligand. The ligand and allosteric site must be compatible with one another, also known as induced fit. When the gate opens, ions such as calcium, sodium, or potassium are able to pass through into or out of the cell. When ions enter, the cell undergoes a change in electrical property which will cause biological responses. The ions that pass through must be specific to the channel and ligand signal. When the cell reaches a point where the concentration of molecules has balanced, the ligand will detach from the allosteric site, ultimately closing the ligand gate. G-protein-linked reception is a way of cell signaling that involves 2 protein components as well as guanine di and triphosphate groups. In the absence of a signal, the components of the G-protein-linked receptors are in an inactive state, where the trimeric protein group, made up of alpha, beta, and gamma subunits, is together and it has a guanine diphosphate group, GDP, bound to the alpha subunit. The alpha and gamma subunits have a lipid anchors that attach them to the plasma membrane. The 7 transmembrane receptors are prepared to undergo a conformational change once a signal is attached. When a ligand bonds at the receptor site of the G protein, it undergoes a shape and structure conformational change that triggers a response from the trimeric protein group. The group then binds with the receptor. Once the group is bound, the activated receptor makes the alpha subunit go through the shape change, which causes the GDP to dissociate out of the subunit. GTP, which is plentiful in the cytosol, binds to the alpha helix to replace the GDP. This new bind causes further conformational change in the trimeric group that separates the alpha subunit and the beta-gamma dimer group to get ready to meet up with target proteins. When the subunits meet up with target proteins, they regulate the activity of proteins by either inhibiting or stimulating. The target proteins then produce signaling to other components which then trigger biological responses in the signaling cascade. Often, adenylyl cyclase is a common target of G proteins, and when activated, it will catalyze the synthesis of ATP to cAMP. Eventually, the subunits will hydrolyze its connection with GTP back to GDP so it can turn back to its inactive state. Often, another protein called the regulator of G protein, RGS, will assist in this process. Once the alpha subunit gets back to GDP molecule, the beta-gamma dimer group will reconnect, the trimeric group will once again become inactive. This means that the process can be done many more times with the G proteins as long as the ligand is bound to the reception site. In this clip, it is demonstrated how the process of cell signaling can be performed multiple times given that a ligand is still attached to the receptor site. Ligand signals can range in various stimuli such as neurotransmitters, hormones, and odorants. G-protein coupled receptors are important in body processes such as metabolism, cell growth, and immune responses. In some cases, the transmembrane receptors will inactivate even when the ligand is still bound to its receptor, in which case a receptor kinase will come and phosphorylate the intracellular membrane area of the receptor. This causes a high affinity for the arrestin protein, which activates the receptor by blocking its interaction with other G proteins. In summary, the connection of a ligand signal will cause changes to the transmembrane receptor and trigger a response from the heterometric protein group. Thank you for watching my video.